Good afternoon to everyone. Our first speaker is Ernamma. Her topic is ultrastructure of plasma membrane and membrane asymmetry. Please share your screen, Ernamma.
Yeah. Hello.
Yeah, hello, Ernamma. Please start your seminar.
One second. Is it visible? Hello.
No, no, it's not visible. Actually, please present your seminar.
Okay. One second.
Please share your screen.
Yeah. Now is it visible?
Yes, please continue.
Okay. Good afternoon, all. My seminar topic is ultrastructure of plasma membrane and its asymmetry. This is a very well-known topic to know us, and I just recall that. First history. The term cell membrane was coined by Sinagali and Kramer in 1855, and the in 1895, Charles Aldrin suggested that the plasma membrane is composed of lipids, and in 1925, he got proposed that the plasma membrane is composed of phospholipid bilayer, and plasma membrane is encloses every type of cell, prokaryotic and eukaryotic cells. Actually, the plasma membrane is physically separate the cytoplasm from surrounding cellular environment, and the thickness of the plasma membrane is 8 to 10 nanometers. And plasma membrane is a dynamic and fluid structure and forms the external boundary of the cells, and it is also act as a selectively permeable membrane. Uh, because of it controls the entry and exit of the molecules or ions. That's why it is called selectively permeable. And there is structure of plasma membrane, and the parts of plasma membrane for extracellular fluids, bilayer, lipid bilayer, channel proteins, glycoproteins, glycolipids, cholesterols. These are the parts of plasma membrane. And the fluid mosaic model is a very acceptable model. And uh, in 1972, Singer and Nicholson proposed the fluid mosaic model, and this is very acceptable model. In this model, membrane are viewed as a quasi-fluid structure. That's why it is fluid mosaic model. And in this, the proteins are inserted into a lipid bilayer, and then the Singer and Nicholson also proposed that the membrane as a dynamic structure, and the plasma membrane is made up of your proteins and phospholipids, and they are also said proteins are alpha globular proteins are present in the plasma membrane, and according to Singer and Nicholson, proteins are two types: extrinsic and intrinsic. I just tell later the types of proteins. And the Singer and Nicholson also tells the ratio of proteins and lipids varies enormously depends upon the cell type. For example, human RBC cell contain 43% of lipids and 49% of proteins, whereas mouse liver cells are 54% of lipids contain and 46% of proteins are contained. That's why the ratio of protein and lipids are varies depends upon the cell type. And lipids and proteins are capable of flip-flop movement. Flip-flop movement is nothing but movement of phospholipids molecule between two layers. That is called flip-flop movement. And there's the components in plasma membrane. And so all the plasma membrane phospholipid. For example, this is the structure of phospholipid, and phospholipid bilayer particle. Yes, this is the structure, and the hydrophilic head and hydrophobic tail are present. And in this hydrophilic head is also called polar head group, and the hydrophobic head is also called non-polar group. And hydrophobic is a two fatty acid chains are present. One is saturated fatty acid, another one is unsaturated fatty acid. In the hydrophilic head, phosphate and glycerol is present. Depend upon the which phospholipid is present. Phospholipids also differ from every cell. And membrane components, proteins are present and lipids, different components, carbohydrates, proteins, lipids are also present in the plasma membrane, and highly proteins and lipids are present in the plasma membrane. That's why it is called mainly made up of proteins and phospholipids. And actually, phospholipid is amphipathic in nature. That's why it is two types: hydrophilic head group and hydrophilic tail is present. That's why it is called amphipathic in nature. And these phospholipid is two types of movement is present. That is transition movement and flip-flop movement. Transition movement is nothing but the movement of phospholipid molecule is same layer. The movement is occur in the same layer, that is called transition movement. Flip-flop movement is the movement of phospholipid molecule between the two layers. It is called flip-flop movement. And these are the phospholipid cholesterols are present, proteins, glycocalyx are also components in the plasma membrane. And these are the types of membrane lipids. For example, glycolipids, phospholipids, sphingolipids are also present. Depend upon the cell type. Which cell type is present, that is that phospholipids are present in the cell. And glycolipids, triglycerol and glycophospholipids, ethyl glycolipids, sphingospholipids, glycolipids, for example. These are the structures, and these are also structures of phospholipid structure. First one is and the sphingomyelin, second one. Glycolipids and sterols are the structures of phospholipids. Carbohydrates, next one of the component, carbohydrate. Carbohydrates are also found in plasma membrane. Most carbohydrates are part of glycoproteins, and glycoproteins play role in interactions between cells. For example, addition. For example, in the addition, the glycoproteins play the main role interaction between the cells. And the proteins are wedged between lipids, which are the molecules that could not pass through ion channels or gates. And proteins in the cell membranes are integral proteins, are also called intrinsic proteins and extrinsic proteins. In the intrinsic proteins are partially or completely embedded in the phospholipid bilayer. Intrinsic proteins are, and extrinsic proteins are these are present or they are attached to the phospholipid head. Extrinsic proteins are attached to the phospholipid head, and these are the loosely attached. And fat soluble proteins are passes through the extrinsic proteins, And water soluble substances are passed through the intrinsic proteins. And the role is cell signaling, cell recognition, enzyme activities are involved in the. These are the different types of proteins present in the plasma membrane: extrinsic proteins and monotropic proteins, multipass integral proteins, single pass integral proteins. Multipass integral proteins are also called beta proteins, beta globular proteins. These are the other which are not present. Uh, PC is nothing but phosphatidylcholine. Choline. PC is 50 to 77% of phosphatidylcholine is present in the plasma membrane, and 80 to 100% of sphingomyelin is present in the plasma membrane, and phosphatidylethylalanine are 87 to 100% is present, and phosphatidylserine is 94 to 100% of present in the plasma membrane. Um, Phospholipid asymmetry in the erythrocyte membrane. This figure is shown that, and the colors are indicate the asymmetric distribution of phospholipids in this picture. Thank you.
Okay, thank you, Ernamma. Now, our next speaker is Pavana Monica. Her topic is viral genetics. Please share your screen. Hello, Pavana Monica. Are you in the meeting?
Good afternoon, everyone. Hello.
Yes, Pavana Monica. Please continue.
Good afternoon, everyone. Let me introduce myself. My name is S. Pavana Monica. I am here to give a seminar about viral genetics. Viral genetics is the study of the mechanisms of heritable informations in viruses, including genome structure, replication, and genetic change. Disease-causing viruses that are particular of interest. However, genetics also use viruses to help understand fundamental mechanisms of molecular genetics. Introduction to virus. Viruses are typically described as a obligatory